All right. Good afternoon or morning or whenever this is reaching you. We're going to have a review session here. Just going to look at some of our older review notes from last big test, our midterm, in preparation for our final. So we'll be kind of covering basically from the beginning of the year all the way until where we last left off. And again, we're going to be taking our test when we get back from all this craziness. So you have plenty of time to study, you have plenty of time to look at all this. But yeah, so we just want to make sure we're not forgetting anything, and since we have so much time to review. So first things first, we have our scientific method here. We always start the scientific method with making an observation, looking at something going on in nature, looking at something just in the world around us, which then should have our curious mind starting to ask questions. What would happen if this happened? What would, if I change this, would this occur? So those are the questions, and we're going to do some background research on that, see if there's already been a study done for those things. If there has not, then you're now more knowledgeable of what's going on, and you can begin your experimentation from there. So from there, we create our hypothesis for your experiment. If I mess with this, then this might happen because blah 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 blah, whatever your research kind of supported. Then of course the fun part, you get to test it out. You get to do the experiments. You get to mix chemicals or whatever your experiment is. Then you analyze some data. You're going to plot some points. You're going to make the graphs, make them nice and pretty. And that's where the numbers will then stand out for you. And then based on your data analysis, you will draw your conclusions. And that's what then you're going to share with the world. Sharing is, in fact, caring, right? So that was what we talked about at the very beginning of the year. And then just in general for this class specifically, biology is the study of life. Bio meaning life, and logi or logos means the study of. So what does it mean to be alive? To be considered alive, you need to have certain characteristics. So you need to be able to reproduce. You need to be able to be made of at least one cell, at least one cell. You could be multicellular like we are, or one cell. You could be either one. Use energy to maintain homeostasis. So you need to gain energy from food or from the sun, and then use that energy to live your life, stay balanced. Grow, organized. So things like organelles, tissues, all those things. We're not just big gooey blobs. There's organization to us. Adapt and evolve and respond to stimuli. So those are the characteristics of living things. So I might be able to say, here are three of the characteristics of living things, and one of them is not characteristic. Can you identify which one is not a characteristic of living things? Then we get into the hierarchy of living things. So going from the smallest little thing all the way to the biggest that we're going to look at. We're going to see things from teeny tiny things like atoms and molecules all the way to the biosphere. So they get increasingly larger and larger. And again, we want to focus on changes. Population and community. That's one that always messes people up. Population in this sense is smaller than a community. So a population is a bunch of the same organisms, and then a community is a bunch of different populations. So a population of deer, then a community of deer, squirrel, trees, lizards. So different communities interacting. Then we get into the ecosystem. It's the different communities with the environment, and then the biosphere, all the ecosystems together. Who coined the term cell? That would be Robert Hooke. He's looking at cork cells under the microscope, came up with the cell theory. There are three, make this a little smaller. So there are three parts to the cell theory. So all cells come from other cells, and I'm going to challenge you to figure out the other two as you're studying. Then we have two main types of cells. There are other types of cells, but for our class, the main two we're going to be concerned with are prokaryotes versus eukaryotes. So prokaryotes are things like bacteria. They're very simple, very small. They have no nucleus. they have single-stranded DNA or sometimes just RNA, and the only organelles they have are ribosomes. That's the only thing they have because they got to make proteins, remember, now that we know transcription and translation. So the only thing they have are ribosomes. Eukaryotic cells, remember, you are eukaryotic cells. That's us, so animals, also plants. They have a nucleus, they have double-stranded DNA, and they have organelles. So they're a lot more complicated, a lot bigger, and can do all the crazy cool fun stuff. I don't know why this is down here. This should have been up higher, but yeah, so this is just how to graph, just going over more about graphing. If you forget, always remember dry mix. So the dependent or responding variable goes on the y-axis, and the manipulated independent variable goes on the x-axis. So please don't forget that. And when you're writing your hypothesis, when you're in that if-then situation, it's if the independent variable, then the dependent variable. So that should help you either craft your hypothesis or would help you create your graph based on your hypothesis. Organelles, we should be familiar with most of them by now. The main ones we're going to get into in a little bit will be the mitochondria and the chloroplast, but please still know some of the other ones, like what does a vacuole do, a lysosome, the ER, and the Golgi. Please be familiar with those. Our four main biomolecules. So now we've been more exposed to proteins. They're super important. They're made from amino acids, which are made by the ribosomes. So that's part of the whole transcription translation situation, right? Lipids are super important. They make up the cell membrane. So when things go in and out of a cell, they're passing through the phospholipid bilayer, or the cell membrane. Nucleic acids, we in transcription translation as well. That's what holds the genetic information. So nucleic acid, so DNA, RNA, that's where we get the nucleic acid. Deoxyribonucleic acid is DNA, and ribonucleic acid is RNA. And likewise in cell energy, when we're eating food or plants are making sugars from the sun, it's about carbohydrates, so storing energy in those sugar molecules. So those are the four major ones. We've now used them in our lesson, so this should be like coming full circle with those topics. Cell transport, just want to know water is going to move in and out of cells depending on the situation. That's called osmosis. So the diffusion of water from a high water concentration to a low water concentration. So from high to low. So in a hypertonic solution, it's really salty. So the water, there's a lot more water on the inside than the outside, so that's why the water goes out. And the opposite would be when it's really watery in a hypotonic solution, it's super watery on the outside and it's not as watery on the inside, so the water wants to go in, and that's when it pops. So hyper, hypo. And then in the middle we have, when it's just right, it's isotonic. So they go in and out at the same rate, so the cell size does not change. Other types of cell transport. So this is technically a diffusion. Make this a little bigger. This is a diffusion, but there's also passive transport, active transport, and facilitated diffusion. So for example, active transport needs energy, like ATP, and that's when it's like pushing the boulder up a hill. It doesn't want to go that way, but it's forcing it to, so it charges some energy. And they have things like where we have the cell membrane and we have these protein channels. So these little, like, think about in Mario, the little pipes that allow molecules that are a little too big to go inside the cell. So that's passive transport through some of those things, or facilitated diffusion if there's other mechanisms like those protein channels. All right. Next we have, why are cells so small? why are they so tiny? Why don't we have big old cells? Why do we have to have such small cells? Why do we have to have so many cells to take care of? And it all comes down to math. It comes down to the surface area to volume ratio. So the surface area compared to the volume. So surface area, if we're talking about like a cube, is 6 times length times width. So it's the area of all 6 sides times, or sorry, compared to the volume, which is the length times width times height. So we got to figure out that ratio, and it really, really wants to have a high surface area to volume ratio. It wants to have more surface area, so more spots on the outside to get nutrients inside that cell, so it can go into the mitochondria or what have you. If it's too big, then it can't get in. So if our cell was like huge compared to this cell, it could get a little bit in, but there's this untouched inside part that's unhappy while the outside part is happy. So we want to make sure we have really small cells. Then we get to the cell cycle, where we have a majority of the time the cell is in what we call, whoops, not that, don't do that. The cell is in, oh no, I'm erasing everything. Oh no, panic. No, I don't want to erase. Go back. There we go. The cell is in what we call interphase. It spends most of its life in interphase where it's preparing to divide. So in interphase, it goes through G1 or gap 1, where the cells just get a little bigger. Goes to S phase, called synthesis, where it's copying the DNA. Then G2, it starts to check for any errors to see if it has any last minute changes it needs to make before it goes through mitosis. So interphase is this whole long blue process here. And then we have mitosis, which is prophase, metaphase, anaphase, telophase. And then the last part, which is kind of its own thing, is cytokinesis. So some people use the expression, I promised myself all the cheese, or I peed myself at the circus, whatever helps you remember. The I is interphase, then prophase is where we have the chromosome starting to form. We get these little X's, and the nucleus is starting to dissolve. Then we have metaphase, where the chromosomes line up in the middle. We have anaphase, where the chromosomes are being pulled apart and they're separating. Telophase, they are kind of going to the opposite sides of the cell, the poles of the cell. The nucleus, the nuclear membrane is starting to reform, starting to come back. And cytokinesis, they've split. It split the cytoplasm into 2 new cells. So the whole point of, get rid of this, the whole point of this is to create 2 identical daughter cells. We want them to be 100% identical. We want to have the same types of cells at the beginning and at the end. We don't want to have random change and variation, because then if you get a cut on your arm, you might start growing, other types of cells like your foot cells, and you start growing a foot on your arm, and we wouldn't want that. That'd be a bummer. So we don't like that. All right, so that's kind of like the first part, the first section. So feel free to rewind, fast forward through all this stuff. This is the same, as you probably guessed, study guide we did before. I just want to kind of walk us through. So this is like the first half, a lot of stuff, I know. And then I just want us to check out, let's get back to the top, one other thing where we have, where did it go? This was our transcription translation. So just to remind us, we have transcription translation is DNA to RNA to protein. So in my example, the DNA was the old recipe for the best chocolate chip cookies. It's the best ever, and it's locked in the museum. We can't take it out. So the DNA is the recipe. The nucleus is the museum. We can't take it out, but what we can do is transcribe our own copy. So you can go in and take a copy of it. And then that copy can leave the museum because it's just a copy, who cares? And then you can take that and change it into the next language of the cookies. So you're going to transcribe the original DNA into so this would be DNA into now RNA. If we want to be super specific, it would be messenger RNA. And then we would make our cookies, and that would be the protein. So the DNA to RNA transcription happens in the nucleus, and the RNA to protein happens outside the nucleus, which means it would have to be in the cytoplasm. And that's what they call the central dogma of biology. So the number one main thing, it's always true, it is the utmost importance. So if we kind of stop talking about cookies for a second, we can see a more what's actually happening in our body. So we have our DNA in our cells, and this protein called RNA synthase is kind of zipping down the DNA, synthesizing or making mRNA, messenger RNA, which then can leave the cell, and it gets latched onto by a ribosome called rRNA. That's what that little r stands for, is ribosome. So it latches on, and every third letter, it looks at it and it matches its opposite three letters and brings in an amino acid. So it matches, these three letters are called codons, and it matches its anticodon to there, bringing it into the ribosome, and then dropping off one amino acid at a time until we have, I don't know, sometimes it's just a few, sometimes it's thousands of amino acids, until it reaches that end where it kind of cuts it off and says stop. Like, let's say it's at here, it'll say stop, and it'll stop the production, and then that amino acid chain will wrap around itself and make a beautiful protein, right? So that is transcription and translation. And of course, those things can get messed with mutation. With mutation. So there are several different types of mutation. I'm going to kind of skip past, this was an example we talked about in class, similar to the cookie example, just slightly different. So with some mutations, we have point mutations, which they change one letter. And that could be, no big deal, but it also could be devastating. It could cause a lot of different diseases are caused by point mutations, and that could cause something that is basically you don't even notice it, and it's a silent, something that it messes up a little bit. In this sense, I think I'm spelling that right. And then one that totally messes everything up, it makes it stop early, or just becomes gibberish or whatever, that is nonsense. So that's a nonsense mutation. So those are point mutations. Then we have what's called frame shift. And that's where we have an insertion or a deletion. And that's where a letter is added in or a letter is taken out of the chain of letters, and that causes everything to slide right or left, and that would then just mess up everything. So if you remember the fat cat ate the rat expression, this would cause one of the letters to be removed and it would make no sense. So I'll just write the fat cat. I'll just do that. The fat cat ate. And I'm going to get rid of this first F, but I'm going to keep the codons together. So it's still the, and then everything's going to shift. So A, T, C, then A, T, A, then T, E. So now instead of saying the fat cat ate, it says the atk, ata, t. So just removing one letter messed everything up, except the word the at the very beginning. So those are mutations, and those are what can cause really big problems or They can actually add new traits when we talk about evolution. That's how we kind of get new things for populations to kind of adapt with and respond to their environment. So not always bad. And sometimes, just like silent mutation, we don't even know that they're there. So this is going to wrap up part one. So stay tuned for part two. So we've stopped now at mutations, and we're going to continue with the next ones next time. All right. Have a great rest of the day. Enjoy studying. Please send me a message on Jupiter if you have any questions. Wash your hands. Stay safe. Miss you guys. This is very strange to do over the iPad. And sorry if this was way too fast or way too slow. It's hard without an audience with this. So I appreciate you staying there with me, and let me know how this went. So when I make part two, if you need any other suggestions. So thank you very much. I'm going to